So I guess it's still officially good morning, so thank you for coming, and let me introduce you to B-MoGen Biotechnologies. There's basically three dimensions of our company, and I'm going to start from the one in the middle, work my way over to the right, and then I'll come back to the one on the left, which is the one that I'm going to focus on. So we are a gene engineering company. We hail from the University of Minnesota, our founders, and we basically have a fee-for-service business that currently is how we kind of make our payroll day in and day out. We kind of hang our hats on all three of these dimensions of our company on the toughest kind of gene engineering challenges that are out there. But we've been growing on our fee-for-service business at about 1,200% for the last 2 years. We're nearly break even for not only our fee-for-service business, but the two R&D aspirations that we have as a company. And we take on the more complex projects, and we can deliver them faster than we think anybody in the peers in our industry. Additionally, over on my right-hand side, I'm guessing it'd be your left-hand side, is we are the first company in the world to actually be able to engineer the mitochondria genome. So we currently are capable of doing deletions in the mitochondria genome, and we're on the cusp of being able to edit the mitochondria genome. So the last area I'd like to focus on is kind of what I want to speak to about today is we have a non-viral gene delivery platform for T therapy where we can deliver CARs and TCRs. We think it's a very disruptive platform, and it can save millions of dollars in the cost of goods sold for these therapies. So when we think about kind of the world, and I came from, my background is I was the Chief Operating Officer of progenitor cell therapy and processed over probably 15 different clinical trials of CAR T therapies, dendritic therapies, and those types of things. And when we think about the genetic modification step that's done in the manufacturing, we think of three big problems when you look at the way that it's done today in the virus-based. First, it costs a significant amount per patient for that genetic modification in a virus-based. There's lack of consistency of those products. There's lot-to-lot variability. When I was at PCT, we actually had 2 clinical trials that had to be stopped because there was failed lentiviral vectors coming in that stopped the trials. And then also the capacity size as we start to think about next-generation CARs that are going to be multiple CARs with switches. We think those are the three big problems. So B-MoGen can answer those problems with our non-viral based method in the fact that we're probably a third to even maybe 25% the cost of a lentiviral vector. Our products are scalable and repeatable. We get the same answer every time we do the genetic modification step. And then our capacity is we've delivered over 12 kb into T cells with our platform. So we're not the only non-viral method out there today, but we compete against Sleeping Beauty and Piggyback. When you look at Sleeping Beauty, it's locked up by a company called Zyopharm that is using that in clinical trials today. It comes from, it is working in the clinic, and it was not an exclusive agreement between MD Anderson and Zyopharm. Piggyback is also currently in the clinic, just entering the clinic. And effectively, it is also locked up by a company called Poseida. So when you look at our method, which is derived from the HAT family, it's a transposon system that we've optimized for CARs and TCRs. It basically is not in the clinic yet, but it is available. So it's not exclusively licensed, and we believe that this is a good platform that we can offer to many therapeutic companies. We have extensive data on kind of comparing our stuff to Sleeping Beauty. We're just building our data set of our comparative results and transfection efficiencies, copy numbers, and those types of things in regards to Piggyback. Additionally, we have the right team to kind of deliver on this technology platform. So when you look at 2 of our founders, the top 2, David Largaspada and Brandon Moriarty, they were part and parcel to Sleeping Beauty moving from the University of Minnesota into MD Anderson's hands and into the clinic. So they were there at the very beginning. Bo Weber, our Chief Technology Officer, is very deeply steeped in IPS and stem cell technologies and editing. I have been with the company now for 2.5 years. At the start of the company, I have a fair amount of life sciences background. As I said, I probably processed over 15 different clinical trials when I was at progenitor cell therapy. And then our CFO comes from Medtronic and Prime Therapeutics. So with that, I will kind of...
Thank you, Jeff, for that introduction. I want to obviously ask about the non-viral gene therapy platform, but before I do that, maybe you can just give a brief comment on the fee-for-service platform that you have, and just touch on, as you look at the therapeutic options that you have going forward, how will that evolve? Is that a continuing business that you plan to invest in and grow as you think about these other possibilities for the company?
Yeah, we do see that as a business that's going to continue to grow for us. We have, from our founders who have been doing this for the last 20 years, we have a lot of know-how that we bring through our company, where we can do things faster and we can do things more complex. As I noted, it not only is growing at 1,200% a year, but effectively funds our R&D aspirations. So we see it as a way to kind of make sure we're optimizing the equity investments that people have invested in the company.
Okay, great. So on the gene delivery platform, I guess talk about where that is. What models have you tested it in preclinically, and how does that stack up against lentiviral approaches, other approaches that we're all more familiar with?
Sure. So we effectively started out with just generic cargo of putting into T cells with our platform and optimized it. We've done a lot of work on taking the transposase mutants to up our transvection efficiencies. Subsequent to them, I'd say for the last 6 months, we've got 3 different generic CARs that we're using and 1 TCR. So we have inserted CARs and TCRs into T cells and into CD34 cells. So that's where we're at currently. And we are working with 4 people just in the last 2 weeks, where we're going to start proof of concept work for them to take their specific therapeutic CAR or TCR and put it into live T cells.
And you mentioned efficiency. Is that where it needs to be already, or is there still work to be done there?
Yeah, so the advantages of the viral vectors are transduction efficiencies, which typically are up in the 50%, and cell viability. So B-MoGen has overcome those 2 particular kind of knocks against DNA transposon methods. We're pushing in the 30% range of our transfection efficiency, and we still have some more tricks up our sleeve. And then on cell viability post-transfection, we're easily over 60-65%.
And is that consistent across cell type that you tested in preclinically? And I'm also curious, you mentioned TCR and CAR-T. Is this a platform that you could apply to different cell types, NK cells, et cetera?
Yeah, absolutely. And our founders have a lot of experience in NK cells, so that's some of the work that we're currently doing. So yes, those are avenues that we're continuing to work on, and it is a platform that can be generically applied.
Okay, so maybe you could just help us crystallize the business model a little bit more. It sounds like you're looking specifically at partnerships to become sort of the manufacturing expert. Would you be looking for essentially an exclusive partner, a major deal to sort of be that hub for one partner, or would this be like non-exclusive agreements that you would look to branch out the scope or the breadth of the platform?
Yeah, our original intention is not to look for one partner, but I obviously never say no. But we do believe that this is a platform that could be broadly applied and help all of the therapeutic companies really significantly reduce their cost of goods sold. So we're either looking to license our technology out, or in many cases, if they don't want to process their cells through a CMO, we could become that CMO because we have the experience in the company to process cells for therapies.
Okay, great. And as you think about, obviously, the ex vivo potential of the platform, what about in vivo? Can you do direct gene delivery? Obviously, liver, eye, some of the common first starting places that we're aware of. Have you thought about that? We
did one initial project that was actually a DARPA award to kind of do in vivo delivery. And at this point, we are still kind of super focused on making sure that just ex vivo delivery is optimized into CAR and T cells. So we plan on coming back to in vivo mechanisms, but right now I would say our platform's not ready for that.
do you have a timeline or any expectations of when you might revisit that, or is that dependent on, I guess, progress made in the ex vivo approach?
It's really dependent on bandwidth. As I indicated, we have 4 companies that are currently trying us on, if you will. And if we're successful with those, we envision that that's going to keep us busy. So we want to make sure we do that work right the first time.
So you also mentioned the mitochondrial DNA editing potential. Maybe you could just comment a bit. I think that's something that's not as well explored by your peers and your competitors. What diseases are applicable there? How far off is that approach from being where current technology is and being in prime time?
Yeah, that's a great question. That's a longer-term initiative for our company, but we see there are certain types of mitochondria diseases, such as Kearns-Sayre syndrome and Lee syndrome, that are where there's deletions in the mitochondria that cause diseases. So we're currently working on making mouse models, so the mouse that have been genetically modified to have those deletions in those areas, so that drugs can be tested on those. And then additionally, as I noted, we think we've made some significant breakthroughs on being able to edit. And there is I'm not sure if I can remember exactly, but there is a disease that will cause people with mitochondria mutations to go blind. And I'm blanking on the name right now, but we think that we would be able to actually correct that genetic mutation and fix that and restore sight.
And just thinking about editing mitochondrial DNA, is it primarily a delivery issue to get into the mitochondria, or is there something about the structure of the DNA that requires unique editing technology? I'm just thinking of your technology versus enzymatic editing technology that we know. Could they not just easily do the same thing and move into that realm if it's promising?
And I'll be the first to admit I'm not the scientist in the room, so I'm gonna kind of attempt to answer that question. But my understanding is when you do a double-strand break of the mitochondria, it dissolves. There is a new technique that we license exclusively from the Mayo that is not a double-strand break, and in order to be able to kind of engineer those particular cells. So we have been able to take what they proved out in a zebrafish and do that in iPS cells.
Okay, that's great. And maybe just help set the stage for the next, call it 12 to 18 months for the company, and talk a bit about the expertise you have with the history at the Institute and gene editing technology that's evolved in Minnesota, and what you've learned from that experience, and what you can do to help leverage that and guide the company forward here in the next year or two?
Yeah, that's again a great question. So as noted, the University of Minnesota is a very steeped gene engineering platform where many things have come out. Sleeping Beauty and Talons were invented there. We hope to be able to basically in 12 to 18 months be in clinical trials with our platform with at least 3 to 4 therapeutic companies demonstrating that it can work just as well as lentiviral at a third of the cost. That's our hope and aspirations for the company, and we think 12 to 18 months is going to take a lot of heavy lifting to get there, but we think we're up for the challenge.
Okay, great. Jeff, well, thanks for your time, and best of luck.
All right, thank you.